Good afternoon. Thank you so much for joining us today. My name is Kelly Book, and I'm a co-organizer of this session along with Anna Nelson. We have a series of wonderful talks prepared for you today, specifically looking at some recent research within mathematical biology, and also really highlighting the way that students and undergraduate students and high school students contribute to our research and make it even better. So I'm really excited to hear these talks with you all today. Before we begin today, I would like to take a moment to address those of us that are not in the room with us today, especially those that did not feel safe coming to the state given the current political climate. I would like to acknowledge the effect of this absence. One, that we're just not a complete community today, and my heart breaks for that. And two, I would like to acknowledge that a disadvantaged community within our community is being further disadvantaged by us all being here today without them. So I would like to challenge you, when in the next few months and next year, when you have given your position the power to enhance the career of someone, I encourage you to use that power to try to undo the damage that we are doing here today by gathering in their absence. With that in mind, I would like to introduce our first speaker we have today with us, Matthew Mizuhara, who is going to be talking about fireflies and neurons and being in sync with undergraduates. Thank you.
All right, thank you so much for being here, and thanks for the organizers setting this up. This is a really exciting session. It's not often we get to talk about stuff like this, right, in this format, so this is really exciting. So I'm going to introduce my collaborators sort of as they come up. You can see a list here of some folks. But actually, I'm going to start in sort of a meta way. I'm actually going to start by discussing a little bit about the structures that exist at my college, at the College of New Jersey, which are really productive for really fostering this really awesome research atmosphere with our students. So a little bit about us. The College of New Jersey, TCNJ, is a public, primarily undergraduate institution with about 7,000 students. So I think that puts us like squarely in the medium range. That's what I consider medium. We're largely New Jersey residents. And our average class sizes are about 21 students. This is from the TCNJ website. So I would say we pride ourselves on having small class sizes. We get to know our students really well. This is important for us. We have really good relationships between faculty and students. And within the Department of Math and Stats, if you look at these numbers, I would say we're a pretty large community given a school of this size, right? I mean, we have 22 full-time faculty and 280 majors. And so those majors are equally split between so about half of those majors are education-focused majors, and the other half are sort of equally split between pure, applied, and then data science stats. Okay, so we have a large community, lots of students, lots of excited folks, and a lot of really active faculty doing lots of really awesome mentorship. And so with that as sort of our backdrop, one of the things I wanted to really highlight is how I even think about bringing on students and how that process works at our school. One of the things I'm super excited about is some new stuff we started doing with how we recruit students. So on the right, not all the headshots of my students. I got the ones I could quickly get from LinkedIn and so on. But a couple of my students I'm excited to be able to talk about. But yeah, we every year do this research interest form now, something that I was really excited to help start a few years ago. And the idea is in the spring, we actually send out information to all of our majors via email, and we have like flyers around the department, but usually the main form is email, and we send out so sort of just the basics of what is research, right? So one of the things is we don't want to assume the students even know that research is a thing. Like I thought back to my experience of, my first research experience was going up to a professor saying, hey, you work in such and such area, I want to work with you. And there was a lot of privilege in me knowing that that was a thing to do, right? And not everyone knows how to do that or is comfortable doing that. And so we've been thinking about ways to break down some of those barriers. So I love this research interest form. Goes out to all the students, tells them what research is, how it's different from being in a classroom, how it's beneficial to not just students going to grad school, but for any career choice, right? I mean, there's a lot of skills you pick up that are transferable. So we'll often accompany this with, we've done lightning talks in the past. Faculty will give 5-minute lectures on their research areas that they're trying to recruit for. Over COVID, we did a website. We made a website that had just like short videos with blurbs about their research. So, and then recently, to faculty, of course, we're all overworked. So, sometimes we just send out blurbs, right? I'm just like, hey, here's something I'm recruiting for. But yeah, so I love this. All the students who are interested fill out this form, goes to a central location. Like right now, it's me usually, right? So I'm like excited about this. So I'm taking all those forms that come in and then trying to pair them up with faculty accordingly. One thing that anecdotally we've noticed, I think, is that we are reaching students this way that we wouldn't necessarily notice as the one who's raising their hand all the time in class, right? We're reaching students, and they're getting an opportunity to start talking to us in a way that we wouldn't have necessarily gotten in a traditional format. So loving that. Again, other ways that we break down barriers, which I really appreciate, is students actually get credit in our major. It counts as one of their courses if they do research. So it's not an extracurricular, it actually counts towards their major. Again, I love this idea. I think our department and our school in general has done a really good job fostering this and really making this a part of the community, right? Enforcing this undergraduate research experience. Once we recruit students, so personally, I usually take on 2 to 3 projects a semester, so usually about 2 to 3 students. I usually start the semester with a mentor-mentee agreement. I'm happy to share that with anyone, by the way, so just contact me. It's not like some magical document. It's just a work in progress that I like to give at the beginning of our research collaboration, just explaining what my expectations are, what their expectations should be. It gives them a chance to tell me what they're hoping to get out of it, what skills they want to maybe build. And again, sort of highlighting for them the differences between research experiences and maybe just a traditional classroom experience. They should not be so afraid of having to go backwards a couple steps and to go forward again, right? It's just trying to demystify that process a little bit. Again, trying to make no assumptions. Meet with students weekly. One thing I've been doing the last few years, which have been really exciting but challenging, is trying to incorporate more group meetings between different projects. So love to hear if other people are thinking about that. Okay, so just over 5 minutes. I have things relatively timed. So that gives you a sense of how I structure my research. So the remainder of the talk, I'm going to give you some examples of projects that I think have been really successful. And so let me give you an overview of sort of my research in general. So a lot of my research nowadays is focused on synchronization in periodic systems. And so I like to think about biological or physical systems that are made up of lots and lots of individual agents that are inherently periodic. And so the 2 examples I'll talk about today are like firefly flashing. So fireflies, right, they're periodic because their lights, let's say, are flashing periodically. Or neurons firing, which, okay, if there's actual neuroscientists here, then I apologize. I'm going to really ruin my description of neuroscience, but we'll think of neurons as just a thing that periodically fires, right? It sends electrical signals periodically. And so, okay, inherently that's fine, but what makes these really cool to me is that when you have a lot of these connected together in a network, or they're all communicating, you can start having some really cool pattern formations. And so the video on the right that you're watching, hopefully you can see, I tried to brighten it, it's always really hard to see because it's so dark, but there is a movie of fireflies that are actually in Southeast Asia, and these are special species of fireflies. Actually, there's some in, I believe, Tennessee as well. They're sort of all over. But they have this nice property that, when they are in a collection together, they will actually start flashing in unison. And that's really, really cool. So they look at the fireflies around them, and they say, well, they're doing that. I'm going to try to mimic what they're doing. And so you get these really, really large-scale pattern formations. You get synchronization. You can even see waves propagating along the tree line there. And it's really cool to see this happen. They believe that this is related to, attracting mates sort of more effectively. And so there's a general question. So my general question is, how do we use mathematical modeling to describe these types of systems? How do we analyze them? How do we learn something about why these pattern formations occur? Okay. The important features today that I'm going to stress are, 1, the role of networks in this. So I do a lot of questions about how does the network connectivity affect the results. And I do a lot with heterogeneity. So there's this conflict between the individuals being heterogeneous, being different, and sort of groupthink, right, wanting to act the same. So that conflict, I think, is very interesting. The first model, which I usually start with my students, and this is sort of historically the first extremely successful model of this, is the Kuramoto model. It's a model of n coupled differential equations. So just briefly, the idea is you take theta I, it's an angle on the unit circle, And why do I want to use the unit circle? I'm thinking about periodic behaviors. So you can imagine just a bead on a ring that's just moving around, right? And if you want to think about fireflies, every time that bead hits 0 angle, then the firefly flashes, okay? So we think about the evolution of those oscillators, those thetas. In these equations, you'll see 2 pieces. One is the omega I, that's the intrinsic frequency, that's my individuality. So if omega I, let's say, is really, really large, that means I'm rotating very quickly. I have a very fast flashing rate. But if omega I is really close to 0, then I'm moving very slowly around the circle. And so that gives me some sense of heterogeneity. And then there's this big sum on the right-hand side, which I will, sort of challenge you to go back and think about in the hotels later. But that sine function, if you do some examples, you'll convince yourself that is exactly the thing you need to make theta I, that's me, want to act like theta j. So if I see theta j somewhere else on a unit circle, that sine function will make me attracted towards them. I want to move whatever direction they are in, right? If they're ahead of me, I'll go towards them. If they're behind me, I'll go backwards. Okay, and I take the sum, so I average those of all of the other oscillators, and I see what happens. That k scalar parameter is basically giving me a way of judging which of those 2 competing forces wins. So to check that this model does what we hope it does, when k is small, you get a simulation that looks roughly like this. Things are uniformly distributed. Omegas are random, say, so you're not expecting any large-scale patterns because things aren't communicating. If k is very small, I'm not attracted. But if k is very large, you do see synchronization events, and so you see things clustering. So that's great. The reason I love this model from a mathematical point of view is it's complex enough to do some really interesting things. It can it can show me different states happening, but it's still simple enough that it's amenable to analysis, and that's sort of where my training comes in. So I have a lot of training in mathematical analysis, and so this is a great starting point. And so, of course, there's a critique that it's maybe not the most biologically motivated thing yet, right? And so one of the things that I like to do with my students is take this model as a starting point and think about how can we modify this to maybe describe a particular system or a particular question that we find interesting. So the first one I promised you was connectivity. And so this is a project I started a few years ago, starting with Monica, and then it continued with Sophie and Youssef. And the question was, well, we want to encode how we think fireflies probably look at each other, which is probably with some sort of distance-based metric, right? So it's not that every firefly looks at every other firefly. It's probably that they look at the fireflies nearby, take into account what they're doing, and then evolve according to that. And so I have a little sketch of that. So I'm the firefly with the question mark, and I look at some radius of things around me. And so we said, okay, well, let's build a model that does that. And so fortunately, other people have done similar things. We could use that for reference. But on the bottom right, here we go. On the bottom right, I have sort of a sketch of what we did. So the model we built was to say, let's put every oscillator on a square lattice. And I'm going to be the center dot here. And so my neighbors, let's say for the sake of today, are the 8 points around me that form the square. Okay, so I see only those neighbors and no one else. And I'll take into account what they do, but no one else does. And then the person next to me or the firefly next to me does the same thing. And if you're curious, I extend periodically, like on a torus. Okay, so for those in the know, well, really what I'm doing here is I'm adding an adjacency matrix with some structure, right? But we can write this into a code, like we can code this up in MATLAB, we can see what happens. And this is a great place for students to start. We can ask my first favorite question, what are the solutions, right? Can we find stable solutions, long-term behaviors? Can we classify them? And so Monica was the first to do this. She wrote some really awesome code, did a lot of exploration, a lot of playing, and she eventually found states like this. And so now, because I'm on a 2D lattice, I want to visualize with color. So the color gradient is given on the right, but I think the important thing here is when you look at this, you see, well, there's like some sort of waves that are propagating, and that sure reminds me, at least I hope it does, of the movie we originally saw where we saw some waves propagating in the tree line. We said, oh, that's really cool that we were able to see something similar. And so we solved this. We call this a 3-2 twisted state because if you take a horizontal cross section, so you go across, you'll see that we wind around the unit circle 3 times. So it's a winding number of 3. But if you go vertically, you'll see you only wind around twice. And so with that, we call it a Q1, Q2 twisted state. And we start asking questions like, well, I found a 3-2. What about 2-1? What about 17-19? right? Can I find these twisted states? Are they stable or are they not stable? So that was the sort of the continuation. After Monica graduated, Sophia took over, started using, actually, because of the amount of data we needed, she started learning how to use our high-performance cluster at our campus and got lots of data. She made this really awesome stability diagram on the right. It's all numerically generated. And now we saw, hey, all those green circles are twisted states that are stable. All the red X's are twisted states which are unstable. And we said, that's awesome. We see something that we can understand. Youssef, actually more recently, has been working on understanding the basins of stability of these equilibria. But one thing that I really wanted to do once we had this numerical result was say, well, can we prove something about this? So it'd be ideal if I could say, well, I have the analysis to show you that this is exactly correct. And so the punchline is, I don't have it exactly, but I'm pretty close. Reasonably close. So we have a theorem that we were able to prove. So you can read the theorem statement, but the picture really tells the story. We have bounds for everything below this dashed line we can prove is stable. Everything above the dotted line we can prove is unstable. Everything in between is a gray zone. And this is really fun because, I mean, the techniques to do this, it's linear algebra, right? We're doing linear stability analysis. And it's just a little bit tricky to get really sharp estimates. And so this is a problem that, you find our paper. I bet you can beat us if you spend some time and play with this and get that gap a little bit closer. So I certainly challenge you to think about that. And our techniques do extend to other ranges of interactions and higher dimensions. But for today, that's where I'm going to leave it. As I transition now to our second topic, I don't have as much time for this one. But that first project was about the role of connectivity. The second one, let me talk a little about that heterogeneity, that randomness that can play a major role here. And so this was really motivated first by neurons. So how do I think about neurons? Again, my very simplistic impression of a neuron is given by this, what was called the theta neuron model. And so this ODE, it's just for a single neuron. For the sake of today, there's two types of neurons in the world. There are excitable neurons, which are neurons where if you leave them alone, they'll just sit at rest. If you kick them enough, then they will fire. And then there are bursting neurons, which don't need any impulse. They'll just fire on their own. Okay. And all that difference depends on omega, which is if it's less than 0 or greater than 0, gives you one or the other situation. Okay, so there's two types of neurons I can describe. And so we said, well, let's put a bunch of them together and make them talk. So how do I do that? Well, I make my Kuramoto model, but you see my term in red is my theta neuron. And so I put a bunch of those together and I make them random. So I have some bursting and some excitable. And I say, what happens when you get them all talking? What's going to happen to the dynamics? And so this was actually an REU project that I mentored Andrew Phillips together. So Georgi and I are collaborators. So Georgi's at Drexel. So he had an REU a couple years ago that we co-mentored some students, including Andrew. And so he was the first one to really start attacking this problem. He coded this up. Again, the first step that we typically take is let's code this all up. He started seeing some interesting behavior. It's a little more complex, right, than we saw before, but we saw that when K is small, that coupling is small, it's a little hard to see, but there's like some sort of behavior here, some sort of oscillations, but it's not so easy to tell what's going on. And then when K became really large, we saw, well, now there's like a really dramatic, okay, now it's a little more dramatic what's going on, like everything's getting pulled around, everything's firing. Kendra, more recently, a student of mine at TCNJ, she just graduated. She took this project, made it even more stark. She made a bimodal distribution version. So now using two colors, you can see those two communities very distinctly. Ooh, the yellow does not come up here. Sorry about that. So there's a yellow community on the right that's kind of wiggling back and forth. The teal community is going all the way around. And so we thought, okay, well, it's nice to understand how these two communities are different. Can we understand the transition from one to the other? It turns out mathematically this is an interesting question because I'm going from one limit cycle to another limit cycle. So, for the sake of time, there's not too much I can say here, but I will say there's some really, really cool analysis that went into this. And one of my favorite parts of this was actually having Andrew lead us in understanding this method. So we took some paper explaining the so-called Ott-Antonsen ansatz. It's a big technique in my field. that I'd never used before this project. And so Andrew was a great sport and led us through this analysis, walked us through the steps that as he was learning it, and we together learned this really authentically. And I've now been able to use it in future things, including working with Kendra. So that's been a really awesome moment for me, is just truly learning something from our students. But the main point is that what it does is takes this really high-dimensional nonlinear system and then turns it into a phase plane problem, which we love phase plane analysis for those who do differential equations. And the thing I'll point out is that we can compare those two situations that we had before as, well, there's small loops on the phase plane that represent sort of the first type of oscillation. And it's a little hard to see, but you have also this loop that actually winds around from going out the left and in the right. So those represent the second type of oscillation. And we were able to describe the bifurcation from one to the other rigorously using some cool techniques. The point of this slide, though, is that we saw this picture on the right and said, oh, you know what? It reminds us of this picture from The Little Prince. And so we affectionately called this our boa constrictor bifurcation. So that's, I think in the paper, it's actually what's called. So in the last, I have 1 minute. Whew. So in my last minute, let me just say, in closing, I mean, what are the objectives of undergraduate research for me personally? I think for me right now, the ultimate goal is student ownership of a problem. The results are nice, papers are nice, but that's not really the goal for me, right? I'm much happier when a student can walk out of my college and go to an interview and say, here's a project I worked on, and let me tell you the techniques I used, and here are the questions that I'm interested in. And so for me to think about that as the main goal, I do find nowadays I'm really thinking really hard about the non-content outcomes equally as much as those content outcomes. Something I need to work on is making my students aware of those as much as I'm aware of it, right? Something I need to continue thinking about is, the importance of communication, or of a presentation, and of grit and all these things. And of course, yes, we love the ability to see stuff outside of classroom experiences, learning analysis techniques they maybe won't see in class. Especially for students going on to grad school, that's particularly helpful. But anyway, I think I'm out of time, so with that, thank you, and my email's there in case you want to reach out. Thank you.
We have time for some questions for our speaker here today.
Yes.
So you said you have 2 to 3 projects. Do you get more applicants than the 2 to 3 projects?
That's a good question. So the question was that I usually work on 2 to 3 projects a semester, but do I often get more than 2 to 3 applicants? So the answer is yes. I often do get more applicants than I can personally handle each semester. One of the things I love about that research interest form is that we are able to sort of figure out how many students we might have in any semester. And if I'm not available, we are fortunate to have a lot of other faculty who can maybe take a student on who has similar interests. So, I mean, one other thing I've been trying to do recently, this past year actually, I had a project that I put 2 students on simultaneously. And really, usually I have them sort of bifurcate at some point. But I think that was helpful too, is, if I'm taking on maybe 3 or 4 students in a semester, trying to keep them on similar projects, not have 4 distinct projects.
Yeah, I guess my question, maybe you answered it already, so because there's enough resources. I think about this a lot in terms of equity, but there's lots of equity concerns, and I've heard people say, oh, just take the best students. And it's like, well, the best students probably are going to be okay,
Right.
right? So I wondered if you had any thoughts or guidelines about how you approach this in terms of figuring out who's the best fit and being equity-minded about this.
Yeah, that's a great question. Yeah, I think the equity piece is really important here, right? I think you're right. And I think it goes back to one of the reasons we started doing that sort of mentorship forum it was really equity driven, right? So yeah, I think so far we've been sort of fortunate that there's enough faculty and enough spots with the faculty that we haven't run into that problem. I think moving forward, if that were to come up, I agree. I think something that we'd have to be thinking about is, like you said, I think it's not necessarily the A students, they will be okay and they'll find opportunities, somehow. And maybe that's not the way we should be judging the most qualified applicant necessarily, right? I totally agree with you there. Yeah.
Other questions? All right, well, thank you for...
Oh, sorry.
I'm sorry. I didn't do that.
No, go for it. Yeah.
Go ahead.
So these are obviously extended projects, but you're saying that you're working with a student for only a semester, or does it summer, or what's the
Yeah.
scope and timeline?
Yeah, so the question was scope and timeline. That's a really good point. It does vary. So some of the students listed here, Andrew, for example, that was just summer REU, so that was, I mean, he did work with us a little bit afterwards, but formally it was really like that 8-10 week period. Some students I've worked with for just one semester. More ideally, I mean, a lot of my students do work with me for a year. I've had students with me for up to 2 years, actually. So I do take that into account in the sense that, you know, I may not have a student for more than a semester, so I do try to find projects that if I know that that's all they have time for in their schedule, then I try to find projects where I think maybe we'll be able to get the wheels on the ground a little bit faster, right? And then students who are with me for 2 years, I'm willing to take a little bit more ambitious projects with them. So, but yeah, I know some of my colleagues really are strict about only having a minimum bound of 1 year, but I actually am generally okay working with students for a semester only, because I understand that it might be tough for their schedule, yeah.
Very good search this time. All right, thanks again.
Okay, thank you.
So a little funny backstory. My wife, Britt Banish, was invited originally, but she's going to Japan next week for a different conference and said, but you could go, so here I am. Sorry she couldn't be here. Acknowledgements to UCO for supporting the trip and the research environment they provide, and also our department, which is really great, our college. There's lots of resources for us and students, and specifically the organizers for putting this together. And I really like the message that we got at the beginning of this session about what it's like to be in Florida right now. And just a quick point, Oklahoma is very similar, except we don't really have the attention, the beaches, and it's hotter. So we sort of understand a lot of the issues here, and it makes being an educator pretty tough sometimes. So I want to give a plug for Katie too. Katie was my first research student. She was at, oh, I was nice here and I didn't put a picture, so you're welcome.
I was not so nice. I put a picture of you.
Oh, great. Little stand-up comedy routine here. Anyway, she was a student at Bryn Mawr, and I was at Swarthmore for a year. And my first semester, I think, I taught a math biology class. And she took the bus twice a week to my class. At the end of the semester, she had to do a research project for her senior thesis. So I started to work with her and her Bryn Mawr advisor, Victor DeMay. And turns out she won the poster prize at the sectional meeting the next year. And here she is today. So goals for this is just to talk about how we involve undergraduates, larger research projects, and the thing that made me really nervous and why I thought Britt would be better at this than I would be is this high quality research element, because that made me and a couple others of us nervous. So what I'm going to try to focus on is sort of the general themes that help people like us do undergraduate research. Some of this is probably not going to be surprising to anybody, but hopefully there's some stuff to take away. So I'm at UCO. We currently have about 13,000 students semester to semester. I've been there since the fall of 2012. We had about 17,000 students at the time. We have lots of support for student research, including a student-initiated grant program. Students can write a 1,000-word research proposal in February. It gets reviewed by a panel of faculty around spring break. They get decisions right after finals week, and then the next year they get research. So they get research funding for 5 hours a week, travel or supply money. So it's a really great program. The timeline makes it tough because they have to plan ahead, rush the day before their proposal's due, and then wait basically 8 months to start their project, but it tends to work well. We also support students on campus for travel to state, local, regional, national conferences like a lot of our schools do. So it's a huge priority for us, like it is for most of you. Since about 2019, kind of coincident with the pandemic, I haven't really worked with math students much, which is why I thought Britt would have been better at this today. I've worked with a lot of college students and specifically a lot of biology master's students lately. I have to fill out my post-tenure review form. It's due in a couple weeks. And I started listing the master's committees I'm on for biology, and I gave up after 7 because I didn't want to think about any more at the moment. But most of my projects are driven by student interest or what my collaborators need. So for me, all of this is really about collaboration. And luckily for me, most of my publications are actually in biology journals, because most of you know that biologists always think we're statisticians. So they're like, hey, can you help me with this data? That's going to come up in a few minutes. And yeah, sure, let's do it. So my department fortunately doesn't care that I publish, biostatistics work in biology journals. I don't publish a lot of math papers, actually. So 2 little short quotes. One of them is from my advisor, the first one. He said, "Never let go of you." So he was really great at collaboration. And at one point I asked him how he did it. And he talked about, settling into all these projects and chipping in where he could. But he encouraged me to not give up what I was interested in. And I mean, that's the one thing that I've totally failed at. So I pretty much let go of me the day I graduated, and now I just work on whatever is interesting. Another influential person said once, it isn't research till it's published. So this sort of got in my head 3 or 4 weeks ago when I was starting this, thinking, well, if it's not published, is it high quality? And I'm going to sort of argue, yeah, so take that. So I'm going to talk about a couple biology students I've worked with recently. I'm going to focus on the math students that Britt has worked with on a couple projects, and then the fact that really programming is this unavoidable thing that is good to embrace. So the theme of this, programming for all, I don't know, sounds stupid, but about 5 years ago I was invited to write a book chapter for this firm series. One of our students who I worked with really closely did an REU with one of the editors of this book and said, hey, your student was really great. Would you like to write a book chapter telling us how you did that work with her? So, well, what do I have to say that anybody is going to want to read or listen to? So I thought about it a lot, and I thought back to the things I'd been doing, and in about 2014, 2015, I did the Pick Math program, which we've heard a little bit about today at different talks. And what I thought about from that was, what do students really need, right? Like, I'm not doing this research for me. I'm mostly doing it for them. So as I thought about it, if they work on something for a semester, they're not going to publish it. A year, they're still probably not going to publish it. So what do they really need? What can they get out of it? And as we talked about this, and Britt joined on to help, math is really hard, and surprise, it's really easy as you're analyzing these models to get stuck in like a page of algebra that is totally useless. And what's the point of that? Like, the modeling process is fun, learning the biology is fun, how it all fits together is really fun, and then you get some really bad algebra problem that's not fun. So the idea is, well, programming is also pretty hard, but you get feedback from the debugger, and you can make little incremental progress that you don't always get from the algebraic analysis. So trying to use one to really drive the other while secretly teaching them R or MATLAB or C++, whatever it might be. So here's the cover of that book. Some of you have probably seen it. I imagine some of you are editors or authors, depending on who's in the room right now. There's our chapter, little link here. A little bit of background on modeling. We featured 3 student projects, toxoplasmosis. Emily is currently a PhD student about to finish at Drexel. Circadian rhythms and the influence of alcohol. Eli is a software developer at Boeing. Influenza and healthcare costs. Chase was a actuarial science major in our math department. So the idea of combining disease and health was interesting to him. He was also going to, at the time, go towards the actuarial track where stochastics was becoming embraced. So we thought a stochastic model would be really cool. So we did a neat stochastic model of this as well. So this chapter features background of those 3 projects and basically a cleaned up version of all the code they wrote and used to do their work. Oh, I forgot to start my timer. Okay. So is this high quality research? I hope so, because they did, Emily went to every conference she possibly could for like 3 straight years presented, gave talks and posters. Great experiences for her. Same for Eli. Chase, not so much on the conference circuit. Emily wrote a Sierra paper, and I think those are indicators of really good research that was high quality to them. One neat thing about our chapter is we put in all the code, so you can basically repeat all their research from the online supplement or the online chapter. So it's kind of an all-inclusive standalone example of what a student research project looks like. All right, so first student I actually want to talk about is Ben. So Ben is a junior biomedical science major. That's what we call our pre-med major. And he had this interest in math, which was expressed to me one night. We were at a college Halloween party, like 7:30 on a Tuesday in 2018. And we found out much later that neither of us wanted to go. But I was standing in the food line, and Ben walks over and says, "Hey, Dr. Laverty, my name's Ben, and I'm in Calc 4 right now. But I really like math, but I'm a biology major. What can I do?" So fortunately, I was teaching a better version of the class that Katie took the next spring, and I said, "Sign up for this class." It was a dream to me to get a biology student in my math biology class, because it's really hard to find a student with that background and interest to take a 4,000 level math class, right? He had planned to go to medical school, maybe graduate school, he wasn't quite sure. But he's done so much with me. In that class, he was really interested in ATP phosphorylation, so he wrote a differential equations model and analyzed that. The thing I'm mostly going to talk about is his capture mark recapture analysis, if anyone knows what that is. At the same time he was doing this research project through this funded program on campus, he was also doing a biology capstone project where he was interested in SARS-CoV-2 binding to cells and the viral entry process. So he was doing all this stuff all at once. And then when he graduated, he didn't have enough. So he wanted to do some stuff with COVID epidemiological data. So we had meetings for the first couple months of summer that year. Just like every Tuesday, we'd get together on Zoom and look at data and stuff. So super cool student. In this project, what he learned is programming data wrangling, which is just a massive part of the job. Data is messy, hard to work with. He took MathStat from me, so he got to see in action some of the class ideas from that class. We read papers on capture mark recapture analysis and applications. And the takeaway from this was a collaboration that was sort of just getting started between me and the biologist at UCL, Michelle Haney, and her collaborator, Franny, at Bethel College. They're mammalogists. They trap mice, rats, voles, and are interested in their ecology and population biology. This is actually me when I was in grad school. My program made us do lab rotations. So I went out to the West Desert of Utah for 3 summers and did pretty intense field work. And the important part of that is you learn how hard biology is and what things are measurable, expensive, impossible. And those are really easy things to overlook as mathematicians. You're like, I need this number. Well, that number doesn't exist. So this mark recapture was, I pitched it to Ben because it was just such a great project for all the reasons I mentioned up here. Fortunately for us, it was a pretty low priority thing for the collaborator. So she wanted it, but didn't need it. So it took a lot of the pressure off of Ben. It's a thing that's really common in field biology to do this. But the software that exists for it, things like Mark and Capture, they have R interfaces as well, tend to be really hard to use. So the first time I did this at UCO with a collaborator, he gave me this stack of paper, and the printer printed out the file names for his analyses at the top. And I don't think it was supposed to, because it was lots of swear words, .text. So this is a thing that people need to do, but it's really hard. It also happens to be this opportunity to synthesize like all the great parts of math, maybe even some linear algebra. So here's a really good reference for this kind of problem if you're interested. It's sort of a hobby project of mine that is nice for the right student. So Ben got a math minor, kind of a super minor. He went to Oklahoma Medical Research Foundation to work right out of school. And because he was going to go that direction, we just wanted him to get his hands on data. We started to do this project reading literature. Really, remediation is not the right word. Just sort of reminding him of all these math concepts and things that would help him do this project. We looked at some small special cases that we could literally write on paper, code. We validated against public data that we could find. And then he wrote his big old script that would fully analyze all this data from our collaborators. So I have a pretty nice script that, as this project continues with the biology lab on campus, we can incorporate their data and continue this. So Ben left behind some great projects. He texted me, oh, too soon. Anyway, here's what it looks like. You get a sequence of captures and non-captures, and you're trying to estimate the probability that you catch an animal if it's there, and that it's there. So a 1 means we caught it. We didn't catch it, so we don't know if it's dead or alive or gone. We didn't catch it in the next season, so we don't know if it's dead or alive or gone. We caught it, so we know it had to be alive. So that gives you some information about how easy it is to capture. You put these together for hundreds or thousands of animals, and you can learn about the life history parameters that are of interest to biologists. Here's the math, the likelihood function. It's kind of neat because he had just been through math stats, so here's exponential functions, talk about exponentially distributed random variables. Here's a little element of the binomial distribution because you're looking at sequences of heads or tails, basically. Messy part over here we don't really want to talk about right now. Do some log transformation because that helps with the coding and the estimation process. So this is sort of the remediation step. Lots of good little algebra details that a typical biology student might not care so much about, might make some mistakes on in their own analysis, but for Ben, this is a really great place to put almost all the math you learned in college together in one place. Oops. Here's what it looked like. We get these likelihood surfaces, parameter estimates, confidence bands around those. And then we sort of had some fun trying to map out home ranges and just learn other things that were biologically interesting, but also good programming. These are capture totals. So I refused to learn ggplot, and Ben was like, I'm going to do ggplot, so I let him, but I didn't help him at all. He texted me on Monday and said, hey, I just got a promotion. I'm a data analyst now. Next week I'm starting the online biostat program at Oklahoma State. When I finish that, I can move up to this level of job. So he's just super happy, super cool. Here's a different poster that he presented, but really exciting. And I think the key is that looking outside the math building is really satisfying. It's good for us, it's good for them, and helps more people than just us. Another quick project, Kobe was a biology student at UCO as an undergraduate. He did a master's. I was really involved with his master's project. He just moved to Mississippi last week to start a PhD program. He was looking at fitting von Bertalanffy curves to a bunch of different fish and looking at the genetic effects, but kind of messy data, lots of different kinds of data, so lots of data wrangling stuff. These projects like this have that have a genetics component. These genetics pipelines are really complicated, and you take the data you get from the lab, put it in the computer, get something out of it. Well, that has to go to some other program, but unless you open an Excel and manually move stuff around, it doesn't work. So these pathways, these pipelines are really pretty hard to do and just super tedious. With a little bit of programming, you can make it much smoother. So we were able to sort of streamline some of their processes. There were undergraduates involved, biology students again, in some of the digitization and calculations that were done to collect the data. So these don't really look like traditional math biology projects, not much by the way of differential equations and stochastic processes or graphs, linear algebra, whatever, but there's just so much going on that these are really satisfying problems that lead to results that help collaborators a lot. Currently working on a couple papers for this. So since Britt was invited, I want to briefly mention what is not my research, but I was recently sort of played a little bit role in helping with some of this. She studies fibrinolysis, which is the breakdown of the fibrin fibers in blood clots. So these turquoise things are fibrin fibers. The little purple dots are platelets, and those things are red blood cells. You get blood clots. They can cause problems if they are in the wrong place or for the wrong amount of time. So her research is about how the fibrin mesh can be degraded with therapeutics and trying to understand the properties of those therapeutics that might make drugs better or safer. Typical student roles for her are things like running experiments in the models that exist, writing code to visualize results in new ways. The sort of more interesting stuff is in the middle, modifying some of the biochemistry, either stochastically or with ODEs, to describe new details in this whole process. Students, as well as Brad, who's over there on the side, have worked recently in optimizing the model performance, rewriting some code to make these big, heavily computational things run a little bit faster. I'm almost done. Bunch of students, they do a bunch of things like modeling in gray, analyzing results, writing code. So these are sort of the skills that we think of, independent of what their actual research was. You might notice a little gap here, 2016 to 19. Real quick, she worked with a bunch of master's students then who did similar stuff, but with a little bit more emphasis on coding and programming. This last little point here is kind of cool. I got involved in the project at this point. For a while, we tried to keep our research lives separate for reasons you might understand. We're a married couple in the same department. But recently, I got involved with these two students and her collaborator. These are both students from biology and bioengineering. So again, not math students, but they're doing research that's really compatible with what we do as mathematicians. So I want to leave this up, I think, for the majority of the question period, which is probably going to have to start like right now. But some of these questions: does research work better one-on-one with a student or in teams? We heard about team-based research a minute ago. I've worked with a bunch of students who aren't math majors. I've also worked with a bunch of math majors. When can they start? If research is going to go to publication, they maybe need to be there for a longer time, which means you have to start them young, but how do you do that? Well, I just learned when I made this slide like 2 days ago, that this firm series has volumes. One is called Research with Undergraduates Before Calculus, and the next one is Research with Undergraduates After Calculus. So advice on how to get students started at different stages. I like my research to be driven by students. When I find somebody that might benefit from research, I say, what are you interested in? With Emily, it was cats. Like, okay, we can make that work. I tend to find my collaborators in biology. I really don't assign students to projects. I just don't find that enjoyable. So I tend to help them with their research. And at this point, that really is my research. I don't really do my own stuff anymore. It's kind of good thing, bad thing. But these are just really life-changing things for students. The whole research process, writing proposals, going to conferences, standing up in rooms like this, sitting in the back of rooms like this. That's it. For us, collaboration is the big deal. It's so much more satisfying. I gotta say one little side story. So I was sitting in the plane in Atlanta, and I turned my phone on, and I get a text message from a friend. It says, leprosy, ... And then somebody else says, just saw, oh geez. So about 10 years ago, I moved to Oklahoma, right? And driving to work, I always see dead armadillos. Like, what's the deal with these dead armadillos? So I start reading about armadillos, and it's like, whoa, they carry leprosy. Well, people in Mississippi, Louisiana, the sort of southern border, get leprosy from hunting and eating armadillos. So I got obsessed with this, although obsession didn't translate to actually doing anything. It's like, oh, that's cool. This summer, I had an intro to graduate research student, and I said, well, I've been thinking about this leprosy thing for a long time if you want to look into it. And she did, and like totally fell in love with it. I land here, and then Central Florida is the hotbed of leprosy, like as of 2 days ago. So I thought that was kind of cool, and this might be enough to make me start to pick up my research, and this is a project cool enough that I might actually assign it to the right student. So I'm going to leave this up because it's kind of the most useful set of things, but if you have any questions, I'll stop.
Hi, everybody. Thank you for being here. Thank you to the organizers. So I'm a postdoc at University of Arizona, and I'm going to be talking about including undergrads in biological fluid structure interaction research. And I'm just going to motivate these problems. So generally, I'm interested in computational fluid dynamics related to biological organisms. And I like to ask questions like, how does their behavior and morphology affect fluid flow, and what purpose does this serve? And so with these two specific organisms, so the pulsing soft coral and the blue blubber jellyfish, these are modal cnidarians. And unlike more stationary cnidarians like the stony corals, which you can see as this before and after picture of this bleaching event, the stationary cnidarians tend to be very sensitive to climate change, and these modal cnidarians really aren't. And so there's kind of an idea here that the fact that they're able to interact with their environment makes them a little bit more resistant to environmental changes. And so the overarching goal here is to have a full fluid structure interaction simulation with biologically descriptive morphology and active tension-driven motion. And I'll kind of dig into what both of those are in just a sec. So I'm going to do the very parsed down motivation of these soft corals. Usually I talk for like 3 whole minutes on this one slide. But these are octocorals that are sessile animals that have algae on them that photosynthesize. So they're similar to stony corals, like in that way. They're also a little bit bigger. But what's different about them is that they actively pulse their tentacles by contracting their muscles. So you can see here in this video, it's experimental fluid dynamics visualization, that they're pulsing their tentacles, and you're seeing this upward fluid flow with these vectors. And so experimental work has actually indicated that the pulsing enhances their symbiotic photosynthesis, so that the pulsing is mixing the fluid around and removing oxygen, which is a waste product of photosynthesis. And so past computational work by myself and others that have worked on this have used the prescribed motion rather than active tension. So with the prescribed motion, you take laboratory data and you prescribe the motion directly. Here, what we're going to do is model the muscle contraction using active tension, and this is really useful because it allows for a more dynamic range of motion that has sort of a biological interpretation to it, as well as you can get these emergent behaviors that you wouldn't be able to get from prescribed motion. Then we also have the blue blubber. So they form, oops, 1 second, the video isn't playing. Well, that's a bummer. There we go. So something that's really interesting about them is they form these large population blooms over short time periods, and this has implications in invasive behavior, as well as a possible sustainable food source for humans. And so in general, jellyfish is very well studied in the computational fluid dynamics community. Our collaborator, Alex Hoover, has also done work with jellyfish with the active tension-driven motion. But typically, people look at jellyfish that don't have very prominent oral arms or tentacles, so they are very focused on the bell. And this is a very reasonable modeling simplification because it makes things much more complicated. And so here, you can see with these blue blubbers that the oral arms, which are these little appendages down here, they're very prominent and they're going to impact the fluid dynamics. So this is really the novelty of this project. And I don't know if it's necessary for this session to really motivate this, but I do want to sort of highlight some of the reasons that I like to work with undergrads. So one thing, and I'm also going to say this is from my perspective as a postdoc in a very research-driven environment. I'm even don't teach very much, like I'm going to end up teaching a lot more later on. But I will say that it does save me time in the long run. So there is this initial time that it takes to train somebody. But I have these large projects and all these other things, and I'm sure everybody here feels this, that you're pulled in all of these different directions, and it can be really hard to actually make progress on one project, much less two at the same time. And so because of the work with these students, I really was able to make progress on this work. And also there's something really nice about the motivation of this, of like, oh, somebody is going to meet with me, and I told them I was going to do something, and then I should definitely do it, right? And then the other thing is that at some point, we didn't know how to do research, right? And somebody was there to guide us, or maybe you're one of the few that like figured it out on your own, but I know that wasn't me. So it's really nice to be able to mentor the next generation of scientists and mathematicians. And something I will say is that graduate school is getting more and more competitive, and so having undergrads that have research experience is really going to give them an edge. And then me personally, I find it to be very fun and rewarding. So here's a little video that I did with the student, Ibrahim Zaki. I'm not going to talk about this project, but we are looking at flow past an idealized prickly pear leaf. Okay, so getting into some of the methodology here, when I'm looking at these kinds of projects, what we end up having is a very complicated fluid structure interaction. So I have these elastic materials that are putting a force on the fluid, and then the fluid is going to be moving these materials. So there's this two-way coupling that needs to occur. And so I use an immersed boundary type approach. So the immersed boundary method was developed by Charles Peskin in the 70s. And then what we use is the immersed boundary finite element method, which is an updated version of the immersed boundary method developed by Boyce Griffith. So to model the fluid motion, we use the Navier-Stokes equations. So we have the fluid density here and the fluid viscosity. And what we're solving for is the fluid velocity and the fluid pressure, and this is on a fixed Cartesian grid. And so this first equation is actually a system of nonlinear PDEs, and this would be the conservation of momentum. The second equation is a conservation of mass, and this gives an incompressibility condition, so that's an assumption of the fluid. And so if we have our elastic material, and normally I have a picture of a coral or a jellyfish, but I'm talking about both, so now I just have like a little, torus. This is not going to be defined on this fixed Cartesian grid. This is going to move and deform separately from this fixed Cartesian grid. So this is going to be this like a Lagrangian frame of reference, and then the fluid is an Eulerian frame of reference. And so a hallmark of the immersed boundary method is using a delta function to reconcile these two different frames of reference. And so here, if we have some force that the elastic material is putting on the fluid, this is defined in this Lagrangian frame of reference, and what we're doing here is we're extrapolating the force onto the Eulerian fluid grid, and then inputting it into the Navier-Stokes equations as a body force. So this is how the fluid is feeling the force of the body. Similarly, we solve these Navier-Stokes equations, and then we have this Eulerian velocity, and then we interpolate that Eulerian velocity onto the Lagrangian boundary, and then update its position accordingly, and that's how the body feels the fluid. And so these two different interaction equations are basically, that we're using is to avoid the fact that we would have to have a moving, deforming boundary condition on a system of nonlinear PDEs. So I would be remiss without talking a little bit about the forces, because I said, okay, we're putting a force on the fluid, but what does that mean? So we are going to use some of the underlying material properties to define this force. So it is going to be related to a stress tensor. So this is the first Piola-Kirchhoff stress tensor, the PK1 stress tensor. And this is shown here in weak form. And so we have a test function here. And so this PK1 stress tensor can be decomposed into passive and active elasticity. And so the passive elasticity is to model the fact that these animals are made out of tissue, and tissue is elastic. And so we're going to model this as a neo-Hookean material, which is nonlinear. For the active tension to model the muscle contraction, we have this tension that's being applied, and this can be decomposed into this Tmax, which is the maximum tension that can be, this is a parameter that we can vary. We have an activation function that's going to model the time-varying behavior of the contraction, so this is something that's going to be applied periodically. And then we also have this spatial function that's modeled to show the location of the musculature. So in this video here, we have white to red. So white means that there's no tension that's applied. And so you can see when the tension is applied that this shows where the musculature is, and then the animal contracts their muscle. And then when the tension is not applied, there's this passive expansion, which is due to the elasticity. So you could kind of think of this as a restoring force, where the elasticity is wanting the body to move back to its resting state. And so these are three-dimensional animals, and we want to have this, be biologically relevant and three-dimensional as well. And this can be really difficult, especially in fluid dynamics, because we have these nonlinear PDEs that we have to solve. And so doing this in serial without any adaptive mesh would take just an absurdly long time, where it almost wouldn't be worth it to even try to run the simulations. And so we're going to use IBAMR, which was built by Boyce Griffith, which is a three-dimensional adaptive parallelizable, can't say that apparently, implementation of the immerse boundary method. And so here we have a need for HPC and using the cluster. And then these simulations are tuned to match experimental data, and then we are trying to observe these emergent behaviors that we get from the active tension muscle model. So we could see things like kinematics and flow structures. And so at this point, you might be thinking, how do you have undergrads be a part of this? So I'm going to talk about a couple of challenges that you may or may not have thought of when I was presenting this. So one thing is that fluid dynamics is a huge field that is, the intro courses are usually not taught to math or biology undergrads, and I do work with bio undergrads. We're also numerically solving a system of nonlinear PDEs, which I'm sure anybody that does computational stuff knows that this is a challenge in itself. We also are doing advanced modeling of fluid structure interaction. So just sort of for context, if you're not in this world, this method is not something that is taught to you until graduate school, maybe. Like I know as a grad student, I wasn't ready for the advanced model. I learned sort of the classic model first, and then it wasn't until postdoc that I was able to get to the updated version here. There's also, just move way forward. There's also an aspect of high performance computing, so students need to have Unix knowledge and how to submit jobs to a cluster, and what does that even mean? And then I also will say that everything that we're working on is academic software. So it's not like industry standard proprietary software like ANSYS, where everything is kind of wrapped all nice in one neat little GUI and you can do all of your analysis and simulations. That's not the case. So we have different software for the model creation and meshing, different software for running the simulations, IBAMR, and different software for visualizing the data. So it's definitely not a trivial thing to include undergrads in this. But I'm going to say, since I'm here, obviously I have undergrads that have worked on this. With these sort of barriers, I've had undergrads that have made very meaningful contributions to both of these projects. So Alyssa Connolly worked on the soft corals, and she just graduated and is applying to grad school this coming fall. And Citlali Ramirez is working on the blue blubber with me currently. And so here are some just overview of the different contributions that they've done on these projects, where they helped create and mesh these models. They ran these HPC simulations, and neither of them had Unix experience or working on Linux at all. They visualized the data, and this is also non-trivial because the data from this ends up being really large. So we're talking like tens to hundreds of gigabytes, and so downloading that is not really feasible. So Alyssa in particular had to download the visualization software on the cluster and run it in parallel and submit like Python scripts. So they not only visualized the data, they also were able to like analyze and interpret the results. And so it wasn't something that was passive where they would come to me and say, okay, what now? What now? I mean, a little bit like that at first, but I kind of slowly was building to, okay, so what do you think this is? Does this look right? What can you tell me about this? And then they were able to sort of at the end of the projects actually do some analysis about the results. And then they both had some element of scientific writing. So Citlali is finishing up an REU this summer, so I'm kind of pushing her to, with her final report as like a preliminary draft of a paper, which I'm hoping that will get published or submitted at least pretty soon. And then Alyssa, I've written a manuscript about the Soft Coral project, and I gave her the manuscript, and she gave me edits, and it was fantastic. Like, she gave me some really good insights and like even corrected my grammar and stuff, which is something that is never something that I'm super great at, so I really appreciate it. And so she's an author on that paper, and she definitely has more than earned her place on that. And so I wanted to talk a little bit about some of these results, since I made everybody sit through all of that math talk. But I'm really going to highlight the contributions that they made to these projects and not talk about myself so much. So here we have a video of soft coral pulsing, and the vectors here are the velocity field, and then the color map is the vorticity. So this is the curl of the velocity field, and you can see that when it's closing, we get these vortices that are forming, and then when it opens, so here we can see these. It takes a second, it rests for a sec. So we get these vortices that are forming, and then when it opens, the fluid starts to rotate in the opposite direction, and it pushes the fluid up and away from the animal, so it's pumping fluid away from it. And so this is very consistent with what we've seen in experimental data, which I think if you saw at the very beginning, that this qualitatively looks really similar, and we did some more quantitative analysis as well. But Alyssa helped me work and tune the parameters so that we were able to really get a sense of whether or not our model was accurately capturing the behavior and the flow structures that we wanted to see from the experimental data. Once we kind of got to this like reference case where we had something that was pretty descriptive, she did some steady state analysis and looked at the volumetric flux of the flow at varying heights, so over here, to see that it was at a periodic steady state. And then she also did a parameter sweep. So again, this wasn't something that I just told her, okay, run these parameters. She really worked with me to even set up the parameter sweeps so that the range of the parameters was going to be appropriate and that the simulations would be appropriate for what we were trying to look at. So then moving on to the blue blubber. This is a little bit of a newer project and a little bit less finalized, but I will say that this visualization and simulation Citlali did all by herself. I did give her a tutorial like about some of the stuff, but this is all her. And so here we can also see the velocity vectors and the vorticity. And I'll just say that we can see these vortices that are interacting with these oral arms, and it's affecting the wake below it. So this is really showing that including these oral arms is going to be important for really understanding the fluid dynamics. And so there was a question of, my initial, when I did this, I just made some oral arms, like, they look close. But we didn't really want to do this. We want something that was biologically descriptive. So Citlali took a bunch of data and videos and some compared this to an experimental paper. And she looked at the oral arms and particular sort of the width and the length in relation to the bell diameter. Because we didn't have any scaling or anything, so she had to kind of dynamically scale this. And then that was this way of sort of looking and making this biologically relevant. She also worked with me on tuning these parameters and some experimental validation. And so here I'm including some of the quantitative experimental validation that we've done. So one of the things that we were tuning for was this maximum contraction percentage, and I'm presenting this in dimensionless form, and just if you're really interested in how we non-dimensionalize this, I can talk about it, but it was so that we can compare this to the biological data because it wasn't clear what size these animals actually were. They gave us a range, and it was like, okay. So anyway, we're getting really, really close with the tuning of this maximum contraction percentage of the bell. But then with the average swimming speed, this is not something that we prescribe. This is an emergent behavior that we're getting just from tuning the contraction percentage. And again, we're getting very, very similar results. So this is just a fantastic first sort of result, and we're going to keep pushing forward and hopefully get something publication worthy at the end of next fall semester. Okay, how much time do I have? Okay. So I wanted to end this with some tips for involving undergraduates in research. And again, I'm going to say that this is based on my experience as a postdoc at our research-focused institution. I'm actually transitioning over to something that's not necessarily a research-focused institution, so we'll see how much these hold up, although I think that there's definitely something to it. So the first thing, and this was actually a suggestion from my postdoc mentor, was to have them work on what you work on and what excites you. This is very much the opposite of everybody else that has talked about this, and I would love to talk to them and hear about how I think maybe this is, maybe being in my postdoc, I'm a little specialized, right? But I'm gonna make a case for this, which is that I think there is a tendency to give undergraduates sort of toy problems. And this completely makes sense because when you first start working on something, you want to kind of work on something that's sort of feasible. But I'll say the practicality of this sometimes can get in the way. So one thing is that, and again, this might be an R1, like a researchy problem, but it tends to not be very publishable. And so if that's something that you're really focused on, you might feel a little bit less motivated to work on it actively with your student. And then the other thing is that you might have just a bunch of projects, and then you might feel a little overextended. So I think this is where this really comes in. And so then you may not be as active as a mentor as that you could be. And then me personally, I love my research, and I think it would be a little bit harder for me to get excited about something that's just like wildly out of my area. But again, other people seem to have no problem with that, so I'd love to talk to you and compare notes. If you're doing some level of computation in your work, I would suggest using open source code, or if you don't have any open source code, try to write your own easy to use or black box code. And this takes a lot of time initially, but the time scale for somebody to learn your code, if you're just writing like a research code, can be like weeks to months before they can even run anything. And so this is difficult for like motivation and also for time scale because there's such a delay between when they can start making meaningful contributions to your research. And so I suggest having this black box style code where they can start contributing meaningfully and then learn the details as they need to. It's kind of like a just-in-time approach in terms of teaching. Another thing that comes on pretty handy for me is having these multiple short video tutorials, sort of like a flipped classroom style. Again, this takes a lot of time initially, but then you have them, and then you can use them for the next person that you're training. And then the last here is mentoring as a two-way street. And so somebody already talked about a mentorship agreement, and I'm going to sort of elaborate on what mine is like. So I have a list that's sort of like what I expect of the students in terms of how much they work a week and their email response times and what research means and all of that. But I also have what they can expect from me. And then I give this to them, and they can read it, and then they can talk to me about it and amend it. And so it really is a mutual agreement that we both have. And I think this is really useful because these sorts of explicit expectations are really helpful for all students, but it's particularly helpful for students that come from underserved backgrounds. And I don't have the citation here, but this has been cited, right? Or actually studied and peer reviewed. The other is that if somebody is not upholding their side of the agreement, and I'm sure that one of the hesitancies of working with undergrads, it seems that from what people I've talked about is they're like, oh, well, what if they just, go do something else, or they start, get busy and then they don't pick up their slack or whatever. And if you both have this agreement, you can sit down and it helps have this potentially uncomfortable conversation, which makes it a little bit easier. And the way I phrase this as well is that sometimes I get busy and sometimes I don't uphold my end of the agreement. So it also helps empower them to be able to talk to me if there is something that's going on. I don't know if I have time. How much time do I have? Okay, so I will just say this last thing here is managing your expectations, not expecting people to know how to do stuff, scaffolding things like research and to your how to do research, how to read scientific papers, formulate hypotheses, scaffold those into your research program, and also balance, giving the details enough so that they can make a meaningful contribution to your work as soon as possible without getting them too bogged down in the details, but giving them the details as they need it. So with that, I'd like to acknowledge my postdoc mentor, Laura Miller, who has just been a fantastic mentor and has allowed me to sort of work on all these great projects. Then Alyssa and Connolly and Citlali Ramirez, of course, they're just wonderful to work with, and I feel very privileged to have worked with such wonderful people. And then Alexander Hoover, who had done all of this great work on this active tension muscle modeling and especially got the coral project off the ground, as well as the NSF. So thank you for listening. Do you have any questions? And I just wanted to, I asked the student if it was okay. I asked for her feedback on my slides, and so she gave me some feedback, which is things that I could have done better, which I really appreciated, and something that I think is really, really meaningful in terms of thinking about how important these kinds of collaborations and sort of opportunities are for people that maybe don't feel like they belong. So thank you.
Thanks, Anna. So first of all, I'm super excited to be here, and Sean talked about being nervous, and I'm really nervous because this is a space that I love to be in, but I have to kind of straddle the industry line and the academic line right now. But I just wanted to introduce you and tell you a tiny bit about me because I've had a non-traditional trajectory. So I was an undergraduate student at Bryn Mawr College, and I was Sean's first student, and we worked on this awesome project that was kind of a combination of both, where I took his class. I didn't know much about math modeling as a junior, and I said, I like the SIR modeling that we were doing, but I also like the intra-host dynamics, but I didn't know that's what it was called. And he's like, you want to learn about really gross worms and a global public health issue? And who knew that now I'm working for a global public health company? So that was back in 2008, and I was co-mentored by Victor Dene, who's wonderful at Bryn Mawr. And so then I moved on, and I went to graduate school not really knowing where I was. And I moved on to work on some viral dynamics modeling at NC State. And my former advisor there, Dr. Banks, who has recently passed away, but he really trained me on some of the fundamentals, and I got to elaborate on math modeling and learn some more quantitative methods on how to analyze the validity and the certainty of models. And then I quit that program, and I just transferred to University of Utah to learn about coagulation, a bit about what Sean was talking about before. And I worked with Dr. Fogelson, and I also got to work with some undergrads there as well. And then I did my postdoc at UC Davis, and this is kind of where the story gets a little murky. And I met my undergraduate, Katerina, who, this is her story, it's not mine. So we'll really dive into that. And that was in 2020, right in the middle of the pandemic. I moved September 2020 to be by myself in a box. Okay, and then I made a completely crazy shift, and now I work at Pfizer with my mentors, CJ Musante, who is also an NC State grad, and then Richard Allen, who is a UNC grad. So it's a very interesting trajectory. But like I said, this is Katarina's story, and she came to me as a sophomore undergraduate who was kind of not sure what she wanted to major in. She's a member of the equestrian team, and she's like, I like horses, I like ag, and I'm currently working in this lab, the Hess lab, but I love math, and I took a math class, and I want to see if I can use math for some of the data that I've been working on. So I kind of live in this happy medium where I steered her mathematically, but she came to me with the problem. So we kind of had the best of both worlds. And I just need to I'm beaming about it because she wrote her own proposal and got funding for her summer project from the Provost Undergraduate Fellowship. She presented in the departmental mathematical biology seminar in the spring of 2021 and did a fantastic job. She was also an intern at different biological laboratories. She did a poster presentation, also at a microbiology conference, and she just completed her undergraduate dissertation in applied mathematics. So, yes, I got her. And now she's also taken a managerial role. So she's the lab manager now at Hess Labs currently. So she drove the manuscript that you can see here that is currently submitted to Frontiers in Microbiology. And I'm not kidding, she drove it. She wrote it. I just, massaged. She's the one who really did most of the writing. So that's Katarina, and I'm going to tell you about her work and how, ironically, working with her really trained me for my current job. So Katarina came to me and she said, I'm working with cows and methane emissions and rumen fermentation. And I was like, cool, tell me more. I don't know anything about it. And she was like, well, it's kind of like why we care about cow births and cow farms, right? So rumen microbial activity is crucial for the animal's metabolism. And it is a byproduct of the fermentation process of the rumen microbes, so the microbes inside of the cow's stomach. And we know that methane emissions, there's a global warming potential that is essentially 28-fold higher than that of carbon dioxide. And so she's like, okay, so this is a problem, and in the Hess lab, we're adding the seaweed to the feed additive to essentially reduce the methane emission. I'm like, what are you talking about seaweed? Why are we adding that to it? And she said that experimental studies have been investigating the impact of adding this ATEX, is how we say it, I struggle with Latin. And it contains this compound called bromoform. And bromoform essentially is reducing that methane emissions. But in its pure form, or in its extracted form, it's carcinogenic, so it's harmful to the cow. So you need to have the seaweed. You need to add the seaweed. And so I'm looking at this, and I'm like, this is a systems biology problem. I feel like there's some complex networks going on. She's like, I got all this data, and I don't know how to analyze it. And what do you do? And it's like, that's what I do, and I love doing that. So we wanted to utilize an extended and calibrated mathematical model to identify some potential targets. So what could we tweak in the system that could also add to reducing the methane? And she was like, great, that's really cool, how do we do that? And so Katarina was a member of, she participated in this entire modeling approach in this cycle. So she came to me with the biological and physical problem. She also was the expert in the experiments and the data collection. And then we kind of broke down the rest of the problem into pieces. So to calibrate and validate the model, we're essentially utilizing the experiments and the data and then kind of communicating them with the model. And then she goes, okay, well, how do we believe it, or how do we know it's true? And she hates that I always say, all models are wrong. And she's like, what, that's scary. And I was like, no, that gives us some wiggle room. So we quantified the model's robustness and we did a sensitivity analysis. And then she was like, well, how are we going to keep Dr. Hess interested? And I said, well, you can tell him that we can identify some key parameters or maybe something about the feed additives that might enhance the methane production. She's like, okay, that makes sense. So that's the changes in our understanding. And then she said, well, are we done then? And I was like, no, we're always iterating. So this is the experiment, and Katerina got me out there in that field collecting data. So essentially you collect fecal matter from the cow, and then you extract rumen fluid as well as like rumen solids. And then we use this fermentation system. So it's called a Rusatec, which you can see on the left here. And we essentially take gas emission data from that fermenter. And specifically, we're looking at methane, and we look at CO2 levels. And then also, we look at the microbiome in that system. So we want to know like what microbes are consisting of that system, and how do they contribute to the gas emissions. And so she was the expert here. She had some of this data. She's like, how can we use that for the model? So we took a published model, and we extended it. So we added in, we essentially added in the microbial contribution to the model, and then we recalibrated it. So as far as getting Katerina off the ground, we spent a lot of time looking at this diagram, understanding the interactions. She did not have ordinary differential equations before she came to me, and I just gave her a 20 ODE system with 50 parameters and a well-mixed assumption, and said, let's go for it. And she's like, oh. And I told her, oh, I also feel the same way. And so the model describes the breakdown of carbohydrates and proteins that is inside of the cow, different production of volatile fatty acids, and then also the production of CO2, liquid gas transfer, and the microbial communities. So like I said before, what's different, what's modulating this system is bromoform, and that's where the little seaweed pictures are. And her job was to interpret the literature of what the hypothesis it was for adding the seaweed additive and seeing how it would modulate the system, and transfer it onto this diagram, like which interaction should we modulate. And so specifically, bromoform directly inhibits methane production, and it indirectly affects interactions through hydrogen control. So I'm not going to talk a ton about it, but those are that we have direct and indirect feedback modulation. Okay, so then she said, well, this model is really complicated. I want to create a diagram, or I want to create a framework in which I understand what's going on. And I was like, me too. Inputs is what she identified. So she said initial concentrations of the bacterial microbes. And then she said the rates of microbial growth and death, the different biochemical parameters, there's physiochemical parameters, and there's also the decay and the source of bromoform, right? We have to add in the seaweed. And then we plug those into our differential equation model. And the solutions that we have at every point in time, right, we're interested in the methane production, which you can see a representative model output there. And so we want to overlay the data with the methane production. And she goes, okay, but we don't have data at every time point. And I was like, no, we don't, but let's see how close we can get. So we use different data for the calibration. And so the question is we can't model every microbe in the microbiome. That would be way too many. So we classified them. We classified them as amino acid utilizers, meaning those microbes eat amino acids. Sugar utilizers, those microbes utilize sugar. And methanogens, so they kind of live off of the methane that's being produced currently in the microbiome. And so that's Katerina's data, which is essentially a relative abundance table of all the different microbes that she identified from the rumen sample. And then we applied the classification, she applied the classification to that type of data to essentially give us some initial conditions for our model. And then what she did from there, she's like, well, what if we modulated those initial conditions? What if there's different combinations. Like we only have one snapshot in time. And I said, I agree. Can we make different virtual microbiomes? And we did. So essentially circled right there are the 3 compartments, the sugar, amino acid, and utilizers, and the methanogens. And so she ran simulations and variations on those initial conditions based off of the original data. And she essentially plotted the distributions of the different methanogens and sugar utilizers and amino acid utilizers. And the dashed black lines are the initial conditions from the model that best fit the methane data. So this was her first attempt at bringing in some variation into the model. So I just mentioned the methane data, and so she fit the model and she calibrated the model with the experimental methane data as well as the CO2 data. So you can see here in squares and triangles are the data from the control, which has no seaweed, and then the bromoform, which has seaweed. And you can see for methane, right, with the control, we have elevated methane, and with the seaweed, we get that reduction in the methane. So she fit the model using an optimization toolbox in MATLAB. She did not know MATLAB originally, and she caught on very quickly. So that was great, and we were really excited about this. And then she said, well, I'm plotting a continuous line on top of these discrete data points. And I was like, yeah. And she goes, well, can I plot observations as a function? Like she wanted to know what it looked like, and if it was linear, like how good the fit was. And I was like, yeah, I agree. And so she plotted on the y-axis the methane observation from the data, and then on the x-axis she plotted the methane prediction. And you get the linear y equals x in this case would mean that we had a really good fit to the data. And I'm really proud of her for that. She did an excellent job. And again, the treatment is on the right, the control is on the left. Okay, that's beautiful. All right, so she said like, okay, I hear you, a modifier of the system, a key parameter. She's like, I don't really know what that looks like or how we're gonna do that. And it's incorporating the statistical analysis and again, that interpretation and comparison of the output. So we kind of drew this diagram and we fleshed it out, and I put some fancy words on there, but this is really Katarina's original diagram you see in red here. And so she's like, well, we have this model, and I want to perturb or I want to add variation to all the parameters, which is going to make a big data set. And then I want to see, what affects the methane the most, right? I want to look at the identification of these modifiers. And then she's like, and then I can go back to Dr. Hess and say, hey, maybe we should try this in the experiment. I said, that would be great. I think that's very exciting. And so she's like, well, how do we run all these simulations? What do we modify? And then how do we quantify this? She told me, she's like, I can't just look at all these curves all day long. And I said, yes, you're right. We cannot look at all these curves all day long. So on the upper right here, we have a methane curve. And we talked about it a long time, and we said we have to take a snapshot in time. What metric are we going to look at? And here you can identify, they have the lag time, or kind of like how long it takes for methane to reach a certain level. And then we also have a max relative rate, so what's the slope of that increase in methane. And then we have the final methane concentration in fluid. And so we talked about different sensitivity analyses. She's like, well, do I vary one parameter or all of them? And I was like, all of them? I don't know. We can do whatever you want. And so we looked at a local sensitivity analysis where she modulated one parameter at a time. And we looked at the difference in the model output over the normalized difference in the model output. We also looked at this thing, which is like a pseudo-global method. It's called the Morris method, and it looks at the mean response and the standard deviation within the simulations that you generate from essentially like a Latin hypercube sampling. And then we also looked at global sensitivity analysis, where we varied multiple parameters at a time, and we could see the interactions between the parameters. And this is Katarina's analysis. So on the top row, that's the result of a global sensitivity analysis, and she's plotting here Sobel indices. So the Sobel indices are essentially trying to say that, you know, the variation in that particular parameter created a large variation in the output. And she goes, I like big bars. And I was like, I do too, that's good. And so she said, well, is it normal to expect big bars that look different between the control and the treatment? And I said, maybe. That's part of our analysis. That's what we're learning. So she identified the parameters that are circled here in both the control and the treatment that modulated the methane production the most. And so that was really exciting. And then in the bottom row, she did her local sensitivity analysis. And the direction of the arrow indicates whether the methane increased or decreased. And then like the level at which it is above 0 is like quantifying the effect. So you could say, for instance, if you increased it a lot, look for the up arrow and it is far from the zero axis. So she did a great job on this. And then the test was, we were going to simulate with those particular parameter changes, and can we make a better and a cleaner version, right? That's really what we wanted to know. And she did this in the control. So if you didn't add the seaweed, are there changes we could make? She identified in the feed composition, so the protein, the carbohydrates, and the non-digestible fiber, changes in the feed composition could lead to a methane reduction. So she's calling this an optimization of the feed itself, not the additive. So you can see here, this is the original, the baseline methane production, and here she was able to get more of a reduction here, adding in those changes to the feed itself. So I was super excited. It was a great project, and she's currently designing experiments to essentially validate that prediction, that model prediction. And she's applying for graduate school in the fall, which I'm also very excited for. And like I said, this is her contact information. She's going to be on the job market soon, / graduate job market soon, and she was fabulous to work with. Okay, about 1 minute. All right. So I also want to acknowledge the contributions from our district, from our biologists. So Dr. Matthias Hess is a microbiologist who also works with animal science at UC Davis, and then his graduate student who really mentored Katarina's wet lab skills is Brianna Rowe, whose data we also use to calibrate the model. Okay, so thank you for your attention. Again, I'm always looking for intern opportunities at Pfizer, and like I said, we made drugs for cows. We can also make drugs for people, and if you have any questions, I love to talk about an opportunity, or if you want somebody to come talk to your students about alternative routes and the skills that they require in an industry job, please let me know. I would love to speak, or I would love to hear their perspective. So like I said, Katarina essentially trained me for my current job based on the fact that we worked on drugs for cows. So now I work on drugs for people. So, thank you.
Any questions for our speaker?
When do we get to read your super cool paper?
When they review it, it's been really slow. So yes, we are working on a version right now. So Matthias, and this is something also to consider when you're working with math undergraduates who are also working in a wet lab. He wanted to submit the paper in the journal first and then put it up on the archive, which I know some people put up on the archive first and then submit. So right now we are polishing off the archive version, so that will be published soon. And then the reviewers, it's been a little bit back and forth, so soon, hopefully. Yeah.
I have a question about timeline, because I know you mentioned Katarina didn't have ODEs.
Yeah.
So what was the time course for this project? How long did you work with her? And
That's a key component. So I worked with her for an entire year. And so I think that's really the benefit here. So she came to me spring of her sophomore year. We worked from summer of her sophomore year until summer of her junior year. And then, I mean, honestly, she kept working with Mateus. And then I would sprinkle in, questions back and forth. So it's been a longer process than a normal RU or any of those types. Yes, question?
Imagining that to feed as many cows as there are would take a lot of seaweed.
That is an excellent point, which is why she...
Would Pfizer then come up with a way of putting this chemical into something else?
Maybe, I don't know. This isn't a Pfizer-sponsored talk. This is a UC Davis-sponsored talk, but I think it's something really interesting to think about. Sorry, I should highlight. I'll go back. Katerina worked for a biology lab, so the Hygeia biological laboratories that also worked on feed additives, and so she was communicating with them. So I agree that upfront cost is a problem, and then also compliance. So ranchers who are working with these animals, trying to get them to be interested in the product or spend the money on the product is also hard. So I think we're a long way away, but I think just seeing the different approaches, and this is kind of where the field is leading, about trying to find different feed additives or different feed compositions.
So if you don't know this,
I
it's
don't.
fine, but so she was looking specifically without the additives and changing the carbohydrates. So is there an assumption that sort of different formulation of the food is still going to be good enough in terms of nutrition?
Yes. So that's what we had Mateus talk to us about, and that was these initial. So the thing is that the changes in the feed composition essentially support a certain microbiome, right? That's more advantageous. But we talked to Mateus a lot about. Sorry, I'm going in the wrong direction. About nutrition and are the cows going to be depleted? Sorry, this is not the right slide. I'm trying to show you this. And so the feed additives are going to shift these distributions too. And so Mateus's answer said, I can only go this low in protein. I can only go this low on carbohydrates. And we use those bounds in our search when we constructed the virtual microbiome. So that's a great question. And that sounds really terrible, but they also have to eat and be fed. So.
Do the cows like the seaweed?
They do. I don't know. Yeah, they do. Yeah, they're fun. So we have two, essentially, that we worked with, and they were very friendly. And Katerina was my ambassador. Even though I went to NC State, an agricultural school, it was the first time I got that close to a cow up in their business. So Katerina is definitely the one who is like, you can get closer. I'm like, no, man. And it was also very beneficial to our working relationship with our biologist because he got a kick out of me in muck boots, like trying to talk to the cow, being like, okay, don't hurt me. We're going to be friends. So, yeah.
It seems like such a great project, but now that you're at Pfizer, are you sad? You mentioned interns, but are you sad to not have? How different is your industry experience from your postdoc?
It's quite a bit different. However, I have been taking interns. So I had an intern this past summer. Carrie Sung, she came from Duke BME, so the biomedical engineering department. We got approval to work on a disease platform model, and then I kind of put my own spin on it. So I have a little bit of leeway about what we can work on. We did some resistance modeling, how antiviral resistance essentially shifts the efficacy of drugs. And I got some bifurcation analysis in there, and the pharmacologists are like, why? And I'm like, because it's important, and I can show you why. So I have a less freedom, and the question is being driven by the organization. But I will say that the students bring a perspective that, like, helps us shape that question in a way that I don't think they were going to go. So, yeah, it's a good book.
Let's thank our speaker again.
Thank you so much. Thank you.
Well, thank you for having me. I don't have to say much about myself now. We can be done. So this is all just different projects I've been working on with my undergraduate students, all related to Clostridioides difficile, looking at different spread within healthcare settings. So just a brief outline of my talk. I will tell you what Clostridioides difficile is. I'm going to show you a mathematical model and result that I did as a graduate student before I became a professor, and then 4 of the projects that have been offshoots of that original work. So Clostridioides difficile, or C. difficile, is just a bacteria that's spread among humans via the fecal-oral route. It'll colonize the large intestine typically after antibiotic use, but the problem is we treat a C. diff infection, again, with antibiotics. So we typically see a 20% chance of recurrent infection, and severe cases will lead to colon perforation and an emergency colectomy, which has an 80% mortality rate. This is the most frequently identified healthcare-acquired infection in the United States. Each year, we see about 250,000 cases and 13,000 deaths. In 2019, the Centers for Disease Control and Prevention ranked C. diff as an urgent threat, which is the highest threat level you can have, in the analysis of antibiotic-resistant drugs, or bacteria, I should say. The interesting thing, at least in my opinion, of C. diff is that it spreads via the environment. So both symptomatic and asymptomatic patients will shed spores that live in the environment for long periods of time, and they're resistant to many commonly used disinfectants. So we often see that transmission will occur when a susceptible patient interacts with a contaminated room that once had a colonized patient. So this was actually the reason that I started a mathematical model, was to incorporate the environment that hadn't been done in other C. diff models before. So we had this paper published in 2021 in Mathematical Biosciences. Lindsay Fox, who was another graduate student at the University of Tennessee, worked with me. She's now at Eckerd College. What we did was we broke the patients into the 4 main classes you typically see. We have R, our resistant individuals who have not had antibiotics. S, are susceptible individuals who have recently had antibiotics. C, are colonized asymptomatic patients, and D, are outwardly diseased symptomatic patients. What we wanted to do when we included the environment was figure out, well, what surfaces lend most to disease spread. So we broke it down into 2 different areas. We have high touch frequency surfaces, which are those that are touched more often. When you contact them, it's for a long period of time, so they're cleaned more thoroughly and more often. Things like bed rails, IV pumps, food trays. Conversely, you have low touch frequency surfaces in a hospital room that you touch less often, typically don't clean them, you're not touching them for a long time. So things like the thermostats, the hand sanitizer dispensers, even the trash bins. So what we did was we explicitly included the density of spores on these 2 surfaces in our model and made it so that these were the only 2 ways that a susceptible patient could become colonized. So we built our 6-dimensional system of ordinary differential equations. We went ahead and simulated it. There's not going to be much of a shocker with any of my graphs. Everything tends to go toward a steady state in our models. So you see here, we have around 16 or 17 susceptible patients at the final time, 5 colonized, 5 resistant, and 1 diseased. When we're looking at our environmental classes over on the right, we can see there's an increase in the density of spores. We're starting these simulations with a fairly clean hospital room. We're ending with a moderate to dirty hospital room, just based on spore density. So this isn't all that interesting. What we're interested in is actually something called incidence. So the way we're reporting incidence is per 1,000 admissions. So when I say that the incidence of colonized, those asymptomatic patients, is 28, I mean I'm going to expect to see 28 patients become colonized asymptomatically for every 1,000 patients who enter the hospital. So we expect around 21 patients to become diseased. The interesting thing with our models, we could see which of the 2 surfaces was leading more toward disease spread. So we saw that approximately 77% of the colonizations came from the high-touch fomites, despite additional cleaning. So those were still leading to disease spread. So I was finishing this up, became a faculty member in August 2021, and immediately thought, all right, how do I get students involved in this? On things that I already know how to do as well. So the one thing I didn't tell you is there's another vector of transmission for C. diff, and it's the healthcare worker hands. So healthcare workers, when they interact with patients, will contaminate their hands, and they can spread that from patient to patient. On top of that, we actually see that hand hygiene methods vary very wildly. Compliance levels can be anywhere from 10 to 90%, depending on the hospital you're in. It averages between 40 and 50%. So just context between patients and healthcare workers can contribute to the spread of infection in healthcare settings. So this was the first idea. I thought, well, how do I get a student involved? We decided to start to include that in a mathematical model. So at Lewis, we have a program over the summer called SURE. I worked with Justina over here on the right, Justina Slowinska. Last summer, I've been working with Kristen S. on the left here this summer, and they've been building a new ODE model. So last summer, Justina was focused on adding this PW class, which is just the seat of spore density on the hands of healthcare workers. This summer, Kristen's been updating some parameter values to make them more accurate, and added in this PD, or the same idea, but the spores on the hands of a doctor. When I say a healthcare worker, for the rest of this talk, I just mean someone who's in close, regular contact with patients, so nurses, aides, type individuals. So you'll notice the schematic looks a lot like my original, but a hell of a lot more complicated. The ODEs feel a lot of the same way. One thing that Justina and I started last summer, Kristen's helped push forward a lot this summer, is trying to decide, well, the healthcare workers and doctors can interact with the surfaces, so how do we want that spore density to transmit? So we've been using this heaviside function here, which is just an on-off function depending on input, which is why we have all of these fancy T's. We just wanted to decide, well, if, say, the high-touch surface had more, so this is the high-touch and healthcare worker interactions. If the high-touch surface had a greater contamination level than the healthcare worker would gain spores, and the high-touch surface would lose, and the same vice versa. So we have all of the different interactions between healthcare workers and surfaces, doctors and surfaces, and even healthcare workers and doctors, in case they were interacting together. So we go ahead, we simulated this model using MATLAB. Notice a lot of these graphs, again, look similar to my original model. The patient classes and the two environmental classes in the bottom match identically to what we had in the original model, but now we have extra details on what's happening with the hands of the healthcare workers and doctors. So you only see a purple line here. It's because the healthcare workers are in yellow, doctors are in purple. Doctors are the same contamination level as healthcare workers in our baseline scenario. So we're seeing that that contamination level is somewhere between low and high touch surfaces, kind of where we expected to see it. But again, we care less about looking at boring graphs and more at, well, what's actually going on with incidence. So now instead of just the high and low touch fomites, we also have the healthcare workers and doctors. And we're seeing that 63% of the colonizations are coming from the hands of the healthcare workers themselves. It's then followed by the high-touch fomites, then the doctors. So those high-touch fomites matter more than the doctors since we have fewer of them in the model, with the low-touch fomites contributing the least. Our total incidence of colonized and disease is around the same as what we saw in the original model. So from there, Kristen has been tasked with trying to figure out, well, how do we make this better? How do we reduce incidence? So the first thing we considered was, what if we varied the way that the healthcare workers and doctors wash their hands? So on the left here, we have our baseline scenario. What we did when we were forming our constants for hand hygiene was we figured out, well, how many spores are killed if you use soap and water, which is 90%, versus if you use an alcohol-based hand rub, which is only 10. And how often are these healthcare workers and doctors using each method? So at baseline, we have around 16% usage of soap and water, the rest being that of an alcohol-based hand rub. And we wanted to figure out, well, what happens if you just use soap and water as frequently as you use alcohol-based hand rub? And you can see that our incidence of colonized goes down by 8 patients, and that's largely coming due to the healthcare worker calculation. Another way we could look at hand hygiene is just at the actual compliance itself. So we were assuming that there was a 50% compliance at baseline. We said, well, what if we just increase compliance to 75%? You only wash your hands 3 out of the 4 times that you should be. And we're seeing, again, around a drop in 9 patients becoming colonized, again, largely from the healthcare workers themselves. There are many other things Kristen's been considering this summer. I don't have a lot of time to go through them all. But she is, in the last week that we have of the program, looking at other ways that we can control and minimize those incidence rates based on a global sensitivity analysis that she ran. And then hopefully in the future, we can get and actually apply some optimal control theory to this model to figure out, well, what levels should we be doing of hand hygiene and other things in the hospital setting. So first project, again, I stuck with the ODEs. The second project I've been working on, I actually did in collaboration with another faculty member at Lewis, Brittany Stevenson, who also happened to go to the University of Tennessee. She also happened to study C. diff, but in a different way. So she focused a lot on agent-based modeling, and I had been very focused on environmental transmission, and we said, well, let's make those two worlds collide. So we applied for an internal grant from the Doherty Center, and was able to hire on 3 amazing students. So we have Malin de la Fuente, Layla Murat, and Maximus Lewis, who are all students at Lewis working on this project. So in case you haven't heard of it, an agent-based model allows us to stochastically simulate the actions and interactions of agents on an actual spatial grid. So what we were doing was simulating the environment of a hospital ward that had 30 rooms always kept at capacity, just like my original ODE model. What we did to differentiate from other agent-based models was include both a high and low-touch contamination level in each room with our patients. We just went ahead and jumped right in and included healthcare workers in the model, so we had both those doctors and nurses, and then we put a contamination level on every room and agent. So we used NetLogo to go ahead and simulate this. On the left here is our GUI for the entire hospital ward. These little 4 blocks represent a patient room, so you can see the different patients are color-coded based on the class so that when we run this, we can have a good idea of what the patients are doing. We included healthcare workers, which are these cute little smiley faces that get angry as they get higher levels of contamination. And they'll only visit the 3 hospital rooms, kind of like a rotation. We have the die up here representing doctors, and again, ways to show contamination level. The doctors are able to visit all 30 patients within this hospital ward. And then we had both the high in red and low in orange surfaces in each room that become darker as their contamination level goes up. Since we could, because we have a spatial grid, we threw on this green healthcare worker station, so when those smiley faces aren't visiting their patients, they go and can contaminate each other in the break room. So again, students had fun building this, but I like to know the incidence results. They know me for this one. Since it's stochastic, every time we run, it'll look different. So I'm reporting for 100 simulations, the mean plus or minus 1 standard deviation. So looking at this, our baseline or our mean for incidence of colonized and disease, they're about normal where I'd expect to see them. The thing that we could not figure out is why the low touch frequency surfaces are the largest contributor to colonizations. This is the only model that we've written so far that's showing that's actually the highest contributor. So the first thing we decided when we started to look at, well, how do we mitigate this spread, is what if we just clean the low touch surfaces daily like we were for the high touch? So if you take a look, we're decreasing the incidence of colonized by nearly 50%, and due to low touch is decreasing by 90%. So there were many single intervention strategies that my students considered. This was the one that decreased the incidence of disease the most, and that was only by 11%. They also wanted to consider, well, what if we do 2 things at once? So we looked at some coupled intervention strategies. So this was looking at hand hygiene compliance levels. So we were looking at, down the rows, the probability to decontaminate for the doctors and across the columns for the healthcare workers. This upper right-hand corner is our baseline. So we could see it, well, how is that incidence of colonized changing as we change 2 different things, at least for the mean? So this is a project we've actually finished up. We're working on writing it up, getting it ready for publication. But as we were finishing this, I'm like, there are so many more things we can do with that. So Brittany and I went for another Doherty grant at Lewis, and we're able to work with 4 amazing research students. So Layla was able to rejoin the team. We started this work in January, along with bringing Matthew Sinise on board. They both got to do fun RUs over the summer. And I got to work with two other really amazing people. We have Ethan Jacobowski and Austin Kind like wind. So they've been pushing this work forward over the summer, and this is something that'll keep going into the fall and spring semesters this year. So we wanted to blow it from one hospital ward to a full hospital. So we decided to do 6 hospital wards to replicate where we have the original data coming from. Matthew and Layla had way too much fun deciding which 6 wards we were going to include, but the reason we wanted these different wards in our hospital was so that we could vary the antibiotic use in each ward, we could vary the patients per healthcare worker, and see how does that affect the spread. So in here, we still have those high and low touch surfaces. Now low is just blue, high is red. We have patients, our healthcare workers stayed the same. We now have the yellow healthcare worker wards. We have a green doctor's ward. We have a pink cafeteria, all these different things you would see in a hospital so that we could really try to replicate it as much as possible. So in our preliminary results, we're not done building. We don't even have doctors in there yet. We're gonna start having the wards interact actually next week once we get back to Chicagoland. But we're able to look at the incidence per award and see what's happening. So the big things that are coming out, at least in our preliminary results, is that psychiatry and and palliative care have the lowest incidence for colonized. This is likely due to that minimal antibiotic use because the ICU has the highest and they're using very heavy antibiotics. The other interesting thing is that the incidence of disease throughout the 6 wards is relatively the same. So we're curious to see if that'll update as we continue to include more things in the mathematical model. So again, we're still working on this. This is very much a work in progress. Things that I'd like to see are just updating the discharge procedures per ward. So we know that in certain wards, you're gonna stay longer. The surgery, you're gonna be in and out fairly quickly. So how will that affect our incidence? And then, again, starting to transfer patients between wards. So what if we have a patient that comes in through the ER, but needs to go into palliative care for some reason? Again, those doctors are to come, and then hopefully we can come up with some other control strategies and see how it changes from just looking at one ward versus the full hospital. So you've heard me talk a lot about hospitals, and I'm like, I can only do hospitals for so long. There are other places that C. diff exists, and the big one is actually in long-term care facilities. So through Puma STEM this summer, I was able to work with Harold Arriga, who's at Lewis University, and Liz Leon, who's at Dominican University, to start actually looking at how can we quantify spread in these long-term care facilities. So in case you don't know what I'm talking about, which is highly possible, long-term care facilities differ from hospitals in just how they function and what they're for. So long-term care facilities really are focused on maintaining the quality of life for residents who have chronic illnesses. These residents typically don't see doctors, not like when you're in a hospital and you're seeing doctors multiple times a day. But unlike a hospital where patients stay in their rooms, residents in a long-term care facility will interact with each other. They'll go to the dining hall. So we see different ways of spread in these facilities. The other major thing is that the average age for a long-term care facility is 85 years old. And just being older age automatically makes you more susceptible to having a C. diff infection. So here we're going to assume that at all times we have no resistant patients. All these residents will be susceptible to disease. So the first thing that Liz and Harold had to work on this summer was what do we even include in this model? So they decided that we would have our susceptible patients, that we would break our infectious class into both the asymptomatic and symptomatic carriers. We wanted to consider a quarantine where if we know that you're sick, maybe we send you into your room, you're not allowed to interact with others, and then also our deceased patients so that we know, well, how bad is this really? The other thing that we wanted to keep in mimicking my original model was that environmental component, looking at the C. diff density within the hospital room. So the way that the different patients can become asymptomatically infected is from interacting with one of the 2 different infectious classes as a patient-to-patient transfer or with those touch surfaces. So again, we build up a system of differential equations. This is where we like to start typically, mimicked very much on my original model, and then wanted to look at incidence rates because that's what I like. The only difference is here, we did a slightly different calculation. That's why the values are so low. Because you don't typically see much admission or discharge into a long-term care facility, we switched to patient days. So you take number of patients times number of days. So these are reported as per 10,000 patient days. So we would expect to see around 5 asymptomatic carriers, or only around 1 symptomatic patient, and less than 1 death, which didn't seem too bad in my opinion. But the interesting thing we were seeing in all of our different strategies that we even started to run preliminarily is that the majority, like always 85% above, of these asymptomatic infections were coming from contact with an asymptomatic carrier. So the surfaces really did not matter nearly as much as they did in the hospital settings. So again, this is a project that just started this summer. We're hoping to keep building this and turn it into other ways of looking at spread in a long-term care facility. Some ideas that Harold and Liz had as they just finished up last Friday, were to start to separate the surfaces into both that high and low touch level, like in my original model, start to include maybe healthcare workers and even visitors that could add to the spread. And then we're hoping to couple that with a model of a hospital so that we can watch how the patients become residents and back and forth. So with that, I have to give a huge shout out to Harold, Molly, Kristen, Ethan, Austin, Liz, Max, Layla, Matthew, and Justina for all their work. I clearly would not be here today without them. They're majoritarily back there. They know how proud I am of them, and I don't want to cry. So, and I also want to thank you all for sticking around this late on a Friday, and I'd be happy to take any of your questions. No, not really. I think there might be small ability, but it's so negligible we wouldn't want to consider it. It really is that, so the spores can live anywhere from at least 1 week to up to a year, depending what surface they're on. So that's why we were focused most on the environmental contamination. That, yes. So I don't think I have to explain that. Yes. Go ahead.
This isn't a mathematical question, more a biology question.
Mm-hmm.
How difficult is identifying this bacteria aside from doing a culture?
Do you mean on a surface or in the feces?
Is there something that would be like the swab test for COVID?
So they would have to do stool sampling to determine whether or not a patient actually has a C. diff infection. It's not very often that hospitals do screening unless they know that they're in an outbreak explicitly. On an environment, it's fairly easy from talking to microbiologists to just swab, put it on a petri dish, and watch the C. diff grow, and even count spores and CFUs. Mm-hmm.
This is an amazing talk.
Thank you.
And as someone who is in rural health, C. diff is a huge problem. So I was just curious about, can you speak to calibrating the model or looking at data, and then have you looked at any access of mobile health, electronic health records, or?
Not the latter. For calibrating the model, we've been focused mostly on, and by we, I mean Kristen, reading. I'm not going to take credit for it. She has the most beautiful binder with all of the notes. So reading a lot of literature papers on what are compliance levels, how do we figure out these rates, how many CFUs, and that's one thing I glossed over. We switched from looking at spores to colony-forming units. I didn't know the difference until this summer. Colony-forming units are the actual viable spores that can cause infection, whereas when we were talking spores, we were just looking at total number of spores, and only around 27% of C. diff spores are CFUs, at least from talking with people. So trying to find parameter values, be able to calibrate the model, make sure it makes sense, and our levels, which everything seems to be fairly accurate. We do have some data back from 2008 from Barnes Jewish Hospital in St. Louis, Missouri, and that helped us with our admission and discharge parameters in the very original model. And some of that dates pre my 2001 or 2021 model as well.
No, that makes sense. drug effect. And that would be really interesting as well, to see, if you accumulate antibacterial resistance to the drug,
Yes.
how is this a mitigation strategy? So, changing the viral component of it, the bacterial component of it would be really interesting.
And that's also if you run into Lindsay Fox, I know she's somewhere here. I don't think she's in this room. I don't see her. She started to look more at antibiotic-resistant drugs as she became a faculty member at Eckerd. So she would be one to talk to more on that, but it would be very interesting to start including in what we've been working on here as well.
Some kind of drug effect, even if it's like phenomenological, to see how it's changed the system.
Definitely.
I have one last question.
Yes.
I'm just really curious how, the scientist or the application is going to feel about the mathematical answers. So, how do you think the hospitals are going to feel when one of your main results is like, don't be nasty and wash your hands?
Haven't tried talking to a hospital yet.
Okay.
I will say, when Justina gave her end of symposium talk last summer, we had someone from the College of Nursing in the room, and she goes, you way overestimated compliance. And we were doing 40% last year. We did up it to 50 this year based on some new released literature papers. And she was the faculty member in the College of Nursing, was main reason I'm like, we need to start including doctors in this model as well, because doctors are 15% compliance on average. I think they recognize that they're pretty bad.
Asking your doctors to wash their hands in front of you.
I was unfortunately watching someone in the ER recently, and the amount of times you go, oh, they didn't wash hands. Crap, they didn't wash hands again. Like, please wash your hands. And I just sit there and cringe. So next time you go to the hospital, I'm very sorry.
For multiple reasons, probably.
Yes.
All right, let's